Good afternoon, everyone. My name is Sasha Forson. I'm the Assistant Director of BioNebraska, and I would like to welcome you to today's webinar, a conversation with Dr. Monica Stoops and Dr. Jessye Wojtusik. Today's recorded event is hosted by Nebraska Women in STEM, an initiative led by Nebraska Cures and BioNebraska. At Nebraska Women in STEM, our mission is to reduce barriers for women in STEM and to create an environment where they can thrive in the state. Part of that mission is bringing you conversations like this one today. Make sure you regularly check our website for upcoming events and our Women in STEM profile series for lots of great stories that are happening right here in Nebraska. And don't forget our upcoming events like the Academia Summit. This will be our inaugural kickoff event. So visit our website, nebraskawomeninstem.com, to learn more. I will now pass things off to my Nebraska Women in STEM colleague and Executive Director of Nebraska Cures, Amanda McGill-Johnson.
Yeah, thank you, Sasha. And we are so super excited to have this opportunity to visit with not just one, but two amazing women in STEM from one of the prides of all Nebraska, the Henry Doorly Zoo and Aquarium. Dr. Monica Stoops is the Director of Reproductive Sciences there, and Dr. Jessye Wojtusik is the lead scientist in reproductive sciences. And, I know I visit the zoo quite a bit, and I often forget that such wonderful research is happening right here in Omaha at the zoo. So quick FYI before I throw it to Dr. Stoops, if you have questions, go ahead and throw them at the chat or just wait till the end. We'll have an opportunity for you to ask those questions. And as somewhere in the middle, we'll also stop for questions. So drop them in the chat or hang on to them for those breaks. All right, with that, Dr. Stoops.
Awesome. Thank you so much. And thanks for having us. I'm just going to kind of provide a basic overview of kind of our division itself and kind of the role that we play at our institution. The title of the talk is From Sea Stars to Elephants, because we definitely run the gamut in terms of the species that we work with. And just talking about some of the challenges of studying endangered wildlife. And a lot of that comes from the fact that in some cases, we don't have a lot of information about basic biology for some of these species. See if I can advance my slides. I'm having some tricky troubles here. I don't know why I'm having such troubles. There we go. Okay. So the main thing I want to talk about first is just our institution itself. So Omaha's Henry Doorly Zoo and Aquarium is one of the few institutions throughout the United States that actually has kind of research divisions associated with it. So there's probably about 5 zoos in the country that have the capacity that our institution has. So we're very fortunate to have that. Mission of Omaha Zoo is to inspire, educate, engage people to serve as lifelong stewards for animals, their habitats, and their conservation. And zoos themselves play a vital role in biodiversity conservation. And in ways we do that are helping to manage the genetic diversity of the many animals that are in our care. When we talk about biodiversity, we're referring to the variety of life on Earth. So that encompasses all living organisms as well as their ecosystems. And we kind of look at 3 levels of biodiversity. So there's genetic diversity, which is one thing we're really going to touch on. Species diversity, so the breadth of the numbers of species, as well as kind of ecosystem diversity. And zoos try and play a role in all of these. One could be through like education by having for instance, like our desert dome or a jungle exhibit, that's really giving you kind of an overview of some of the ecosystem diversity that goes on, as well as kind of the breadth of the species that are there. The Reproductive Sciences Department resides in the Center for Conservation and Research. So we're a small but mighty building. And that makes up the departments of animal health, reproductive sciences, conservation genetics, nutritional sciences, as well as animal behavior and well-being. So kind of the approach we're taking is has been referred to as kind of like One Health. We kind of take that in terms of like the animals that we're managing. We try to take it from like that holistic or global approach to managing our animals on a breadth of levels, not just the physiology, but also the behavior. So our Department of Reproductive Sciences, we help to support the maintenance of healthy and sustainable populations. So we develop and conduct research to expand our understanding of the physiology of many species. And that's to make sure that we have sustainable management of our populations. So it doesn't mean that we just have them now. We're trying to have it so that we have healthy populations moving forward for like greater than 100 years. And it's important to have this just for individual animals' well-being, but also for the species in general. Some of the ways that we do this is we can help to facilitate natural reproduction. So we can help to time breeding introductions. We can help to determine the reproductive potential of individual animals. In some cases, we're understanding whether there are distinct breeding seasons that might occur. And in most cases, we are trying to have individual animals breed naturally because we want them to be the species that they are. But in some cases, that may not be able to be done. And so we need to develop some of the skill set that can help to do assisted propagation, which might involve sperm cryopreservation, artificial insemination, and in vitro fertilization. A lot of the challenges that come with research in general, but in this field, we have in a lot of instances a very limited sample size. When you consider, let's say, Philippine crocodiles are one crocodile species that I work with, and there are less than 150 of those animals in the wild, let alone the number that are in managed settings. So getting information and gathering information, sometimes you have a very limited number of animals that you're working with. Some cases we have little to no background information on those species or their reproductive biology. In several instances, we can use model species. So let's say for like a rhino, closest living domestic relative would be a horse. So we can look to that industry where there has been a lot of information gained in terms of reproductive biology due to the economic impact that that species has in terms of the racing industry, as well as just the pleasure riding industry. But again, there can be some similarities, but a lot of big differences between the species. And then there's logistical issues, laws, regulations, the Wildlife Trade Act, as well as limited funding. And I think that's hitting a lot of places, especially not just academic institutions, also government institutions, as well as ours in terms of some of the things that are happening right now with grants and funding. So this slide's just to kind of show, again, in a perfect sense, we want to have animals breed naturally, rhinos be rhinos, tigers be tigers. And if we can help to facilitate that, either through some of the technology that we use, or if we have to take some of the more advanced techniques. Some of the techniques that we do utilize include endocrine analysis or hormone analysis, ultrasonography, exogenous hormone administration, gamete collection, so that could be eggs, sperm, embryo freezing. One thing that we have moved into in terms of rhino species is doing sperm sorting. So sorting sperm for X or Y chromosome so that we can do artificial insemination to produce female calves so that we can better manage our populations and have them reflect more of what it is in the wild, where it tends to be one male with multiple females. And we always like to stress too, we don't want to get rid of the boys. We just want to have a select number of boys for a population, just because of how we manage those species and how they have to be managed individually. Because in many cases, you can't have them as a group together. Males and females in some species only come together for breeding purposes. And then, of course, artificial insemination is one avenue that can be utilized as well. So in terms of endocrinology, this is an area that's pretty near and dear to my heart, kind of what I was classically trained in, just the study of hormones. It's very useful for timing, pairing of individuals, telling the reproductive status. Is an animal cycling? Are they pregnant? Are they not cycling at all? And in the instance of wildlife species, we tend to try to use non-invasively collected samples. So that could be urine samples, fecal samples, saliva samples. We've even used nasal secretions and gotten hormones out of it. I have in there a shed skin because we've done hormone analysis from rattlesnake sheds to be able to look at some of the dynamics of our exhibit animals. So in terms of reproduction, we can tell a lot from the hormones. We can also tell a lot about just kind of the well-being status of the individual animals as well. So we might be looking at something of how an animal utilizes an exhibit or time of day or things that might be going on. And we might look at adrenal hormones for that to figure out if there's any correlation. Hormones, we think about, there's progesterone, estrogen, testosterone, but that's kind of just the basic native hormone. Wildlife species, they excrete many different types of metabolites of hormones. And I have a little slide here that's just showing some representative profiles. When we talk about elephants, the top left graph that's there is showing cycles from elephant and then a pregnancy profile. And again, that's a 22-month pregnancy, so a very long gestation. They have long estrous cycles. They're about 13 to up to 18 weeks, so they only have a few cycles each year. And they excrete just naturally. They don't excrete a lot of native progesterone. They excrete a particular metabolite of progesterone. And so we have to have a specific antibody for that particular metabolite, which is a 5-alpha reduced pregnane. And so that's stuff that you kind of, as you work with species and new species, you're trying to figure out what hormones are they producing? Do we have the matrix to be able to measure the hormone? Do we have the right antibody to be able to do it? So we have to validate our assays for most of the species that we work with. Top right is showing you a hormone profile from one of our female giraffe. We were confirming pregnancy at that point. So that was being used to help with management wise to know that the female had conceived. And so we were able to then bring the male out of the group and have her go on to gestate and produce a calf. Bottom left is showing you one of our elephants. If you don't know, we've been very successful with elephant breeding here at Omaha with 5 calves on the ground and doing very well. We monitored their pregnancies all throughout. This graph is showing you the end of gestation. And we're able to collect samples, monitor them on a daily basis to then know when an individual is going to give birth. So usually about 2 to 5 days before birth, the progesterone will start to decline. And so we're able to then use that to let our animal health team know, our animal care team know, so that they can be as prepared as possible. And this was especially important because we have taken the approach of managing our elephants as a herd. And so we were having herd births occur and making sure that we had the individuals together for that purpose. On the bottom right is an ocelot. This is a species, like many cat species are, where they actually are induced to ovulate. So we see the little blue blips that are going on there. Those are all of her estrogen cycles there. So she has estrous cycles. And then once she bred, she then ovulated. And we have that red showing, which is progesterone. And she actually conceived. And then she gave birth to a single cub who's very healthy. And he left this past fall to go down to Oklahoma City. So we kind of get this little window into their world just through some of those urine and feces and all that other good stuff. One of the other things, semen collection and cryopreservation. When we talked about kind of biodiversity as well as genetic diversity, we have within our reproductive sciences department about 20 liquid nitrogen dewars that contain genetic material. In many cases, it's sperm that's frozen down from a variety of species. And that allows us to maintain that genetic potential of an individual that may no longer be within that population. Ways that we obtain these samples are through a process that's used like in domestic livestock, through either electroejaculation, urethral catheterization, and there's work going on now. Dr. Jessye is leading a project with looking at some chemical induction of obtaining samples. So in a lot of ways, we're trying to have the ability to collect good samples with the least impact on the animal, as well as having it accessible to more people being able to do this, because there's so few of us that are out there doing some of these things, that the more that we can train others or have approaches that are more accessible for veterinary staff or other animal care staff to be able to do. Cryopreservation does require a lot of differences, like we talked about with endocrinology, optimizing that for a particular species, regulatory considerations, and then again, moving toward that area of being more field friendly. Last thing I'm going to touch on is ultrasound. It's very useful for monitoring follicular development, determining sex, and diagnosing pregnancy. Two common methods that are used are transabdominal. A lot of people are familiar with that. If you've ever had to go in and have, let's say, a pregnancy ultrasound, you would have that done transabdominally. For many species, we can do that to diagnose pregnancy. Our animal care teams work really hard with the individual animals, with that bond that they have, to be able to train them for these procedures so that they're doing it voluntarily. One example that you see at the top is an aardvark, and that is a little tiny aardvark within the uterus of that female, and that was obtained transabdominally. For rhino species, which weigh up to 6,000 pounds, they have very thick skin. We generally cannot diagnose pregnancy until after about 100 days of gestation by transabdominal means. So what we tend to do is what is used in the livestock industry with horses and cattle is we do transrectal ultrasound. And so rhinos are trained to be able to have this procedure done, and we're able to view the reproductive tract. One of the images here, this kind of composite, is just showing you again the breadth of kind of species differences of the rhino species that are listed here. There's two Asian rhino species, the Sumatran as well as the greater one-horn rhino. And then there's two African species, the African black and African white. And we have a huge difference in terms of their pre-ovulatory follicle size. So Sumatran rhinos, they grow about a 19 to 20 millimeter follicle, and they're actually a species that is induced to ovulate. African black and African white rhinos, they grow follicles that are very similar in size to a domestic mare. So they're about 45 millimeters to 35 millimeters. And then we got greater one-horn rhinos, who like to go big and proud with everything. And they grow really large follicles. So their normative follicle size that they're going to ovulate can get up to 200 millimeters. So that's kind of like a softball size that's present there. And that's totally normal for them. But again, that big breadth of difference between the species is pretty fascinating, and it really excites us in terms of finding out some of this new information. And I think I'm going to end there and let Jessye take you down a little deeper into some of the individual species that she's working with.
Great. Thank you, Dr. Stoops. Real quick, does anyone have any questions after that overview? And then we'll dig into some of the species. Give folks a moment. You want to go ahead and take down your slides and change the slides while we give folks the opportunity. Otherwise, Dr. Wojtusik, go ahead. You're muted. There
Good
we go.
catch. Hi, everyone. My name is Jessye Wojtusik. I'm the lead scientist in the Reproductive Sciences Department at the Omaha Zoo. I'm going to follow up on some of what Monica talked about, which is some of the tools and techniques that we use to promote conservation in via reproductive sciences, but have kind of a more species-specific take on it. So the two species that you're looking at here are the sunflower sea star and the common hippopotamus. And I will tell you a little bit about each of those throughout this presentation. So the first species I'm going to share with you is the sunflower sea star. This species historically ranged from Alaska all the way down to Mexico. Unfortunately, in the early 2010s or so, about 90% of the population was wiped out by a wasting disease, and this disease continues to threaten the stars that are still surviving. And I do want to give a quick heads up if you don't want to see what's sort of like a graphic picture of what stars look like when they have the wasting disease, please look away now. I'll let you know when to look back. So you can see in this picture, this is a star that has experienced wasting. Unfortunately, it's really devastating. It ends up killing the star. And it's just a horrific, terrible disease. Okay, you can look back, it's safe now. So why are we interested in the sunflower sea stars beyond the just terrible disease and how it's impacting them? They play a major role in the ecosystem of the kelp forest. And if the kelp is known as the sequoias of the sea, the sea stars are known as the wolves of the sea. They play a role of a top predator, helping to manage the urchin population. Without their participation in managing urchin populations, we end up with an overpopulation of urchins, which will decimate the kelp forests and lead to the creation of barrens, which is essentially deserts within our oceans. And this really limits the benefit to the environment that the kelp forests have. Kelp forests are one of the most productive ecosystems, and they are responsible for things like sequestering carbon, as well as buffering the shore from storms, and they provide habitat to a wide variety of species. So we really want to help maintain those ecosystems by helping maintain the balance of the species that reside within them. So from a reproductive sciences perspective, we saw an opportunity to help through the use of cryopreservation. Sea stars are broadcast spawners. So what you see in this video over here is a sea star broadcast spawning millions of eggs. So they are seasonal spawners. All those little white specks that you see are eggs floating in the water. And there's just millions of them upon release. So we can collect these eggs as well as the sperm, which is similarly broadcasted. And our goal is twofold. So our first goal is to cryopreserve the sperm. This is a somewhat simple, easy technique that we use frequently. And the purpose of freezing the sperm is so that we can share those genetics with our fellow aquariums. So if they have a female star that is spawning, they can use that sperm to create larvae to increase the numbers of the population. Our second goal is to use in vitro culture to create larvae. So we will combine collected eggs with collected sperm, allow them to fertilize and create larvae, which we will then attempt to freeze. This allows us to send frozen larvae to, again, a number of aquariums or potentially in the future be used for reintroduction. And so facilities that do not have adult animals in their care could raise sea stars from a larval stage. So this is a general overview of the development of the sea stars following fertilization. This first step, oh, sorry about the volume. This first step is called a gastrula. You can see a small invagination that will eventually develop into the digestive tract or the gastric tracts, hence the name gastrula. The second photograph you see is just further development where you can see that invagination has gone in further. Eventually, when these start to feed, that will change color based on the color of the algae that they're eating, which is pretty cool to see. You'll see little either red or green inside their little gastric system. The first two stages are the stages that we're working to freeze. The more complex the entity that you're working with, the harder it is to freeze. So we're working at a very basic level. These individual gastrula are very, very tiny and very simple in their makeup. So they're a little bit easier, but we still have a number of challenges facing us as we attempt to freeze them. The following stages, you can see they become more alien looking in the middle stage, which is when they are proper larvae. They develop these long tendrils that help them to move around mostly. Eventually, those tendrils will get reabsorbed and then they'll start to take on the shape of what looks more like an actual sea star to us. And finally, they'll grow into a tiny juvenile sea star in this number 5 photo. So we are working with a number of other institutions on this project. This is not an individual project. There are more organizations that I could include on the slide. So we're going to talk about it as the umbrella organization, which is our SAFE program, which is monitored through the AZA. This SAFE program was created to bring together multiple institutions to address the challenges that endangered species are experiencing. So there are a number of species that are within SAFE programs. If you have an interest in hearing more about that, I encourage you to check out the AZA website where you can find all of the SAFE species there and learn more about their programs. Within the Sunflower Sea Star SAFE, or the SSSS, which is insane. We've had a number of successes already. We're a very young group, but we've had success in cryopreservation of both sperm and larvae. We've had a group has worked to identify markers of sex so that we can distinguish males from females. And also we've had some work done towards the identification and the treatment of the disease that is causing the wasting in this species. There has been some successes in that. We're hoping to expand on all of those. In the meantime, we also have a number of goals that are related to the development of identification and monitoring tools so we can check in on these stars as they're surviving in the wild, and looking at the identification of genetic markers for resilience to disease. So our ultimate goal is reintroduction based on genetics, ideally animals that are resilient to the wasting disease, and hopefully recovery of the kelp forest. So that we can come back from the urchin barrens, which you can see up in this top photo, just a completely decimated ecosystem, to healthy ecosystems like you can see in this bottom photo. So now we're moving on to a much larger species, the common hippo, which is also known as the Nile hippo. And this has to be one of my favorite photos ever. It's just very representative of their feistiness, I think. I'm going to tell you a mini love story of when BB met Henry. BB and Henry were introduced after a breeding recommendation was made based on their pedigree genetics to contribute to the maintenance of the healthy hippo population. And we were interested in a way to develop a technique for first diagnosing pregnancy, as well as monitoring fetal development upon pregnancy diagnosis. So our two main choice methods were hormone monitoring and ultrasonography, but we had two challenges that we needed to work around. Challenge number one, we often work to measure hormones in feces as it's a non-invasive biological material. And for most species, it's relatively easy to collect. And I don't know if you know this about hippos, but this is an example of how they defecate. So we were interested in finding an alternative potential biological material for measuring hormones because this is a little bit difficult to clear out of the water that they are usually near. Our second challenge is that hippos are genuinely feisty, and we were a little bit worried about the logistics around ultrasound and whether they would choose to participate in such an exam. Oh, there's the video again. To address the first challenge, we decided to validate an assay to measure hormones in saliva instead of the feces. We used a tube on the end of a syringe. And the care staff worked with BB to develop the skill to have her open her mouth on command so that we could collect the saliva that pools in her lower lip. And this worked a treat. It's important when you are validating assays to make sure that they're biologically relevant or demonstrating the patterns that would be expected based on biology norms. And so what you're seeing in this first graph up above, where you have your peaks and your troughs about every 30 days or so, that's a normal cyclical pattern that we would expect to see for progesterone during the non-pregnant phase. In the second graph, you're seeing elevated progesterone throughout a pregnancy to the point of birth, which it drops back down to normal levels. Also something that we'd expect to see. So we were able to say that salivary progestogens were demonstrating the typical patterns that we expect. We also compared saliva versus fecal samples that we were able to collect and were able to demonstrate similar patterns, showing that it is a good alternative to feces if it is easier to collect. In the meantime, the care staff was working on operant conditioning with BB for her to be willing to participate in ultrasound exams, transabdominal ultrasound exams specifically. And it turns out she's very highly motivated by food. So we were able to conduct ultrasounds on a bi-weekly or weekly basis, as long as she was given her favorite snacks in return. So you can see a couple pictures of her. The top left is showing her getting her reward for standing quietly, and then on the bottom you can see two angles of the ultrasound exam, which was a really awesome experience. So on day 79 of the pregnancy, a typical gestation in common hippos is 8 months. We were able to see the first images of a fetus, and this was the first time that this had ever been done in this species. And just so you know what you're looking at, I added a couple of artistic renditions just to give you a general idea. Later on, we were able to see a beating heart. So if you watch this top, you can see the actual movement of the heart valve of the fetus in utero. And here, this bottom image is of the ribs and spine. And so this eventually all led to the cutest baby hippo being born. Her name is Fiona. And Fiona was actually born prematurely. She was premature by about 8 weeks, and she has a pretty amazing story. If you have any interest, if you Google Fiona, you will find her because the city of Cincinnati is obsessed. Rightfully so. There was a lot of care that went into her survival in her first few months, and so she's very well loved there. But ultimately, we were able to contribute to the maintenance of a healthy individual and a healthy population, which is our ultimate goal from the start. And that is the end of my presentation. So thank you all for listening and happy to take any questions.
Yeah, how awesome, and just what remarkable work you do that is kind of like maybe a hidden secret of the zoo. Um, and like even just thinking about the sea star and the kelp forest and how important that is to the ecosystem and the coastline. Like, thank you for giving us this look behind the curtain of what's happening, at our zoo. Folks, if you have questions, go ahead and pop them in the chat. For now, I'm going to pivot a little bit and just ask each of you about what got you interested in this. Where did your STEM journey begin, and how did you get to where you are? Dr. Stoops, do you want to start with that?
Sure. Let's see. My STEM journey, I guess, it started very early in love of animals and a love of finding out what happens when like something else happens, like trying to figure out the process. And I was somebody that I actually didn't think I would go further than like high school. So I certainly went a lot further than that. And that was just because I think I hit the right field and what I was very interested in. I got my bachelor's in wildlife and fisheries, master's in animal science, and PhD in physiology. And so most of my work prior to coming into the zoo field had been in free ranging animals. So mainly working with elk species and ones that were just living on the land. And then I had wanted to get managed animal experience. And so I did a postdoc at Cincinnati Zoo and then was hired on after that and just became totally fascinated with pretty much any animal that has gonads, which is pretty much every animal. And so they're kind of fair game. And I'm obsessed with all the creatures that we work with because they always teach me something just about them as a species, but also just the fascinating physiology behind them.
What then brought you to Omaha?
Omaha, I came here in 2019, and I was very interested in the fact of I had worked previously here through some of my grant work with rhinos. And so I knew of the rhino collection that was here, that there are two of the species, two of the three managed rhino species here, and very interested in the fact that there was room for growth in terms of the department. So as we talked about, the field is very small in terms of zoo research. That generally means positions are very limited as well. And so having the ability to take more of a role of growing a department and being able to have kind of maybe that impact was very important.
That's really cool. And Dr. Wojtusik, I'll switch over to you now. Tell me about your journey.
Sure. So I grew up on a farm. We had cows and pigs and sheep, and I participated in 4-H. So I knew really early on that I wanted to do something with animals. Wasn't sure what that would look like. My undergraduate degree is in animal science, and that started helping me narrow down my points of interest, which were reproduction as well as conservation. And so I started reaching out to different zoos to see, and researchers that were working on projects that were of interest to me, and just trying to figure out how I could meld those two fields. I eventually I still did my master's in animal science, specifically looking at chickens, which of all things, which is very different from wildlife. But my PhD was focused on wild hoof stocks. So um, primarily the Adragazelle, which is a gazelle from the Sahelo-Saharan region, and there's about 100 left of them in the wild. So we're working with a highly endangered species. Um, which was just really eye-opening to me and just a complete different tact to the um, like domestic species. So you're just it was kind of a great learning experience, learning how to do research with one type and then the other type, because you're just getting a whole different holistic view of how to approach things. I also did postdoc at Cincinnati, focused on iron overload disease and rhinos, and then worked with polar bears for a postdoc for a while. And then I came to Omaha.
Cool. Well, thank you both for sharing a little bit about yourselves. We have a couple of questions now that someone submitted in the chat. First, have you faced any barriers studying reproduction, especially as women? I've heard from other reproductive scientists that this kind of science can be off-putting to some people just because we're dealing with sex, and as a young scientist, it sometimes worries me how I portray my science. So any thoughts? Either of you want to take that question?
Sure. I'll say that if there is an inappropriate joke or similar, we've heard it many times. Um, I think that I don't know if I have advice necessarily as how to approach it, but I've always been really proud of what we do and the things that we're achieving, and what the overall messaging is. And I think it's also an opportunity to educate people. I find that a lot of people, when you talk about what you do, are more interested in it once you get past the awkward part of it. So if you can admit that, yeah, it's kind of awkward and it's kind of funny, and like once you get past that part, that it's actually really cool, it gets easier over time.
That's awesome. Are there a lot of men, or are they mainly women in this space in terms of researchers?
I would say in terms of researchers, it's primarily women, but when it comes to the roles that we work with closely, whether that's upper management or whether it's animal care, it's more biased in the other direction.
Interesting. Huh. And then there was a follow-up question from the same person. What are your processes for finding methods that work for each of your species? She's works with spiders down there in Dr. Hebbet's lab at UNL. So thoughts on that?
We love spiders. Um, yeah, that's awesome. We have a researcher actually here that is working with the group at UNL, is interested in doing work with tarantulas. And so tarantula reproduction. And so that's a novel area. And in terms of the species, a lot of times it's like we talked about, there may be a model species that we work with. So for exotic cats, it might be a domestic cat where it's a model species. In other cases, it's just kind of, you learn a lot also from the keeping staff, the animal care staff who really have that relationship with the animal. We could have some species that they may not show very overt signs of being in heat or in estrus, but an individual person that works with that animal every day might notice this little nuance of like, oh, they didn't eat their grain this day. And that's the cue that it is for that individual. So we rely a lot on that connection. And then a lot of it is just trial and error of working through it. I know I started, one of my main projects when I started out was to develop artificial insemination for rhinos, which had not been done before. And so I learned very early on that instead of looking at kind of the end goal, is looking at those little wins that happen along the way. So one, just like, what is the reproductive tract like in the rhino, and can we get through the cervix, and how do we do that, or how do we collect sperm from the rhino? Because a normal probe that we would use on other hoofstock species doesn't work. So it's developing some of those tools and techniques by just kind of that hands-on.
Cool. And there was a follow-up question that I think was just sent directly to me about hippos and how the training factors of getting them to cooperate. How do you get them to open their mouths so you can collect that saliva, right?
Yeah, so our teams, the animal care teams are really great, and they are very well trained in training. I am not. My dogs don't listen to me at all. So I I would love to put you in touch with people that know how to train. I think primarily it has to do with like operant conditioning, catching them in the act and rewarding the behavior that you're looking for. So if they happen to open their mouth, saying something like yes or good, or and giving them their kudos, whatever that may be, if they're food motivated or whatever. But honestly, I feel terrible trying to speak for the trainers. So if you I'm happy to put you in
People
touch.
know what they're doing. Another follow-up question from someone who I think may have joined us late, so you may have covered it, but may need to say it again. But you talked about the pedigree genetics used with hippos. Is there any sequencing being done with these animals to get a more fine-tuned look at their genetics, or is it not necessary with what you're trying to accomplish?
Yeah, so we do aim to use high-level genetics for managing our populations. The challenge that comes with that is the cost, and like the people time resources. So we use pedigree as the default. It's not ideal, but there are instances where we are using greater genetic tools to manage our populations, but not for all species, certainly.
I'll give folks one more chance. If you have any final questions, put those in the chat. But I also want to ask both of you if you have any advice for women in STEM, or if you've struggled through anything as women that you would like to share, as long as some advice.
That's a good one. Yeah. I think it's one, don't doubt yourself. That's the biggest, is know your own worth. Because you may not ever hear that from an outsider, but recognize your own worth, your own wins. And I think the biggest thing too is like, I know I get the biggest charge out of working with teams. So being able to work as a bigger group to accomplish something, because it isn't just one person or something that's doing it. It takes a lot of people, and sometimes that doesn't always get across because it's always like this flashy, things happened, but like all the steps that go into it. I guess that would be mine, is teamwork makes the dream work.
Love it. Jessye, do you have anything?
Yeah, I mean, I agree with Monica, the know your worth, that it can be really difficult sometimes, when you know what you're doing is important, but it's not necessarily seen that way by others. I think that's super important. I also think it's important. I think this topic came up at the Nebraska Women in STEM conference, actually, the idea that you aren't your job. And I struggle with that a lot because there is a huge identity portion to working in conservation, and the idea that, well, if I don't do it, who's going to do it, and who will save these animals? But you can't let your job get in the way of the rest of your life, and that can be a real struggle. So I don't have advice there though, because I still struggle with that. So working through it, but I think it's important to keep in mind that there are things outside of work.
We're all doing the best we can, right? And making progress and being able to see those things and then just be mindful of them. That's actually really incredibly good advice. Sasha, do you have any questions for them?
No, I think this was awesome. Appreciate you both very much.
Thank
Yeah.
you for having us.
Yeah.
Are there any final thoughts you would like to share about why your work is so important?
I think that part of it is that we're all working so closely with so many different organizations, so many different teams, so many different people. Um, Bringing everybody together for a common goal that is beneficial to the rest of the world on some level, whether they know it or not. I think that keeps me going. I really like the people aspect of it, as much maybe as the animal aspect of it.
Cool. Dr.
Very
Stoops?
well said. Oh, pretty much the same. Yeah, it's definitely I feel very fortunate to work with the team that I have. I feel very fortunate to have seen people grow in this field because it's a hard field. It's a challenging one, again, given the size and the opportunities. And so for me, knowing that there are some good people, you know, that are out there and taking part in it um, is gives me some hope.
Wonderful. Well, thank you both for taking the time to share your information and just what you do with our network this afternoon. And thank you everyone for joining us. Look in the chat for some thanks there. I'm also dropping a link to the survey that we do after all of our events. It'll only take you like 1 minute. Just let us know what you want to be learning after this from future webinars, future speakers, and just enjoy the rest of your day. Thanks, everybody.
Thank you.